My name is Bakish Hamina binti Adam, 97298 from Resource of Biotechnology.
I'm Magdalene Tenkawe. My metric number is 97500 and I'm from Resource Biotechnology.
My name is Dania Karenisa binti Madani, 97367. I'm from Resource Biotechnology program.
My name is Valerie Coalition, 97858 metric number. I'm from Resource Biotechnology.
I'm Greta Yo Yun Qian, 97428 metric number, and I'm from Resource Biotechnology.
Introduction. The 3,5-dinitrosalicylate acid method for estimating the concentration of reducing sugars in a sample. Reducing sugars contain free carbonyl group which have the property to reduce many of the reagent. When alkaline solution of 3,5-dinitrosalicylate acid reacts with reducing sugar, it is converted into 3-amino-5-nitrosalicylate acid with orange color. In this experiment, the spectrophotometer was used to determine the absorbance at 575 nanometer and concentration of sugar in a sample. Spectrophotometer is a method to measure how much chemical substance absorbs the light by measuring the intensity of light as a beam of light passes through sample solution. This measurement correlates to the amount of light that is absorbed, transmitted, or reflected at a specific wavelength through the sample. With the spectrophotometer, the amount of known substance can be determined by measuring the intensity of light detected.
The materials used in Practical 4 are test tubes and test tube rack, distilled water, cuvettes, micropipettes and tips, glass beaker and hot plate. Selected drinks act as the sample, and spectrophotometer.
Practical 4 method. 6 clean and dry test tubes are prepared. 1 ml DNS reagent is added into all test tubes containing samples as measured according to the below table. Third, the test tubes are boiled for 5 to 7 minutes. After boiling, let the test tube cool down to room temperature. Fifth, 1 ml of roach-set salt solution is added into the mixture. Lastly, the color development of the mixture is then read at 575 nanometer by using a spectrophotometer.
In conclusion, this experiment familiarizes with the DNS method for quantifying sugar concentration in a given sample. Throughout the experiment, we learned the significance of this method in accurately determining the sugar content by reading under 540 nanometer using a spectrophotometer. By following the procedure, we get experience in handling reagents, conducting precise measurements, and interpreting calorimetric results. DNS as a reagent can detect and quantify reducing sugars, offering a reliable means of assessing sugar concentration in experimental samples. It was designed to determine sugar concentration in a given sample by employing a glucose standard graph. The utilization of a standard graph allows for a quantitative assessment of sugar content. This not only showcases the significance of calibration, but also equips us with practical skills applicable to analytical techniques.
For this practical, we learned that we are getting used to handling micropipette as our previous experiments also required us to use it. Second, our comprehension of the methods used and the significance of accuracy in biochemical assays was enhanced by contrasting the outcomes with standard solutions that had known quantities of sugar. This experiment made clear how important precise analytical methods are to scientific research and quality control procedures, underscoring the importance of paying close attention to every little detail. We also facing some difficulties, which the micropipette that we use for the whole experiment were contaminated with a yellow solution, which can lead to inaccuracy of the data for spectrophotometry.